Hello, good afternoon, everyone, and thank you for joining us for the Kirby Institute seminar series today. So I'd like to begin by acknowledging the traditional custodians of the land on which we gather today. For me in the north, that's the Woolloomooloomoolong land, and pay my respects to their elders past and present. I extend that respect to the Aboriginal and Torres Strait Islanders peoples joining us here today. The format for the seminar today will start with 2 presentations, followed by a question and answer at the end. So to ask a question, please click on the icon with the speech bubble and question mark in the top right-hand corner to open up the chat panel. And please remember to write your name, as this will help us to reference your question and make answering easier. So now I'd like to introduce our first speaker today, Katherine Ognenovska, an HDI candidate in my RNAi silencing group in IVPP at the Kirby Institute, and she has just submitted her PhD thesis. So Katherine will be sharing some very exciting data from her thesis on identifying the RNA-induced transcriptional gene silencing machinery of an HIV block and lock therapeutic. Thanks, Katherine.
So I thought I'd just start off with acknowledging the Gadigal people of the Eora Nation and pay my respects to their elders past, present and emerging. So as we all know, one of the greatest challenges to finding a HIV cure is the latent reservoir. Strategies have been employed to try and find a cure. Predominantly, they can be divided into 2 major categories. The first is sterilizing. As the name suggests, it's based on the idea of completely purging the virus from the host system, usually through the use of latency reversing agents. One well-known strategy is the shock and kill, which has been highlighted in box C. The second major category is a functional cure, which can be defined as controlling the viral reservoir without its eradication and without art. One approach is box D, where you block and lock the virus through strategies such as, and this RNA interference will be the strategy that will be the focus of this talk. RNA interference can be categorized into 2 major pathways. The first is post-transcriptional gene silencing, or PTGS. It uses short interfering RNA complementary to the target sequence to find and degrade messenger RNA. Transcriptional gene silencing, or TGS, on the other hand, uses siRNA to target genomic DNA and induce chromatin compaction. So in regards to HIV, it can lock the provirus into a latent state. This process is also known as epigenetic silencing, and I'll be referring to it as such from here on out. Now, our lab has developed an siRNA called PROM-A. We've shown that by targeting the conserved NF-kappa B motifs in the promoter region, that we can highly suppress the virus. As such, it has potential as a functional HIV cure, with the end goal being gene therapy. However, while we know that PROM-A induces epigenetic silencing, we don't know the underlying mechanisms. If we can elucidate the pathway, then we could use that knowledge to enhance siRNA therapeutics. When it comes to humans, our knowledge is very limited. We know that in the cytoplasm, our siRNA co-localizes with a protein called Argonaut-1, or AGO1 for short, and together they're trafficked into the nucleus. We know that sequence complementarity is essential for this nuclear entry step, and that siRNA with no genomic target are denied entry. This has led to the idea of the gatekeeper, which is a mechanism that has been shown in other species. What's responsible for this mechanism in humans has yet to be discovered. Finally, we know that siRNA form RITs, an RNA-induced transcriptional silencing, but we don't know what other proteins are part of this complex. If we can shed some light on this pathway, then we could potentially use that knowledge to devise delivery strategies, enhance RNA design, and increase the overall safety and efficacy. Now, we know that AGO1 is part of the human RITs complex. So in theory, if we extracted AGO1 from our cells, then the RITs complex should come with it. Easy, right? Well, AGO1 has been implicated in 40 different pathways and interacts with a large number of proteins. So how do we know what we're looking for? To get an idea, we can look at SPOMB. This fission yeast is a great basic model for cellular processes. It's simple in that it only has 1 copy of key regulatory proteins. It also shares many chromatin modifications with higher organisms such as humans, so its knowledge can be partially transferable. It's for these reasons that many researchers have used SPOMB to study epigenetic silencing, and the process, which is highly characterized, is actually a positive feedback loop. So just briefly, if we look at A, we can see that the siRNA are loaded onto AGO1 in the Argonaut siRNA chaperone complex, or ARC. ARC is a quality control mechanism that ensures that siRNA loading precedes RITs assembly. When required, ARB1 and ARB2 dissociate, and are replaced by CHIP1 and TAS3. To form a fully functional RITS complex, AGO1 cleaves and removes the passenger strand in the duplex RNA. Both ARC and RITS are assembled by chaperone proteins. To induce heterochromatin formation, RITS is targeted to promoter-associated transcripts through homologous base pairing. Once the target has been identified, CHIP1 anchors RITS to the chromatin by a methylated histone 3 lysine 9. To methylate the surrounding region, RITS recruits the CLRC complex, which contains a methyltransferase. CHIP2 and SWE6 promote heterochromatin spreading by recruiting the HDAC complex SHREC. If we look at D, then we can see that SWE6 also recruits the RDRC, which uses the promoter-associated transcript as a template to synthesize double-stranded RNA, which is then cleaved into duplex siRNAs, thus creating a positive feedback loop. Now, from the diagram, we can see that there are quite a few proteins and complexes involved in the overall process. But out of all of them, the RITS complex is the one that's considered the driving force. So for this project, we decided to focus primarily on the human RITS complex, with the hope that identifying those proteins would allow us to branch out and identify the rest. Now, what makes this process so interesting and also challenging is if we look at the yeast RITS complex, then we can see that there are no apparent mammalian homologs of CHIP1 and TAS3. So unfortunately, we can't use sequence homology as an indicator for human RITS, but what we can do is use functional homology. If we dissect the yeast RITS complex, then we can see that AGO1 is responsible for binding and cleaving the duplex siRNA. Human AGO1 isn't catalytic, so already we know that we need a protein with splicing activity. Whether it's part of human RITS or an accessory protein remains to be seen. When it comes to CHIP1, its role is to anchor the complex to methylated histone 3 lysine 9 via its chromodomain. And finally, TAS3 is the bridge between AGO1 and CHIP1. The schematic on the right highlights the binding domains. So now that we have an idea of what we're looking for, we proceeded to extract the human RITS complex. First, we prepared the cells by infecting them with HIV and then transfecting them with PROME. The idea being that this treatment would flood the cells with active RITS complexes. 48 hours after transfection, we extracted the cytoplasm, nucleosoluble, and nucleally insoluble proteins as 3 separate fractions. We then trialed 3 different methods of pulling down AGO1. The first 2 were indirect and used either a flag or GFP tag, whereas the third directly targeted the protein. For all 3 methods, we tested 2 different elution conditions, and mass spectrometry was then performed on the eluate. This was not a trivial process. It actually took years of optimization, and method A was conducted before my time by Dr. Chantal Allensteel and Dr. David Van Buckel. Now, remember how earlier on I mentioned the gatekeeper? Combined with the yeast ARC complex, these concepts suggest that there's a preliminary or additional complex in the cytoplasm. Whether they're also capable of entering the nucleus is unclear, but to try and simplify the process as much as possible, we decided to focus primarily on the nuclear fractions, as they are the ones that, in theory, should consist of fully functional RITS complexes. So for our analysis, we asked, what proteins are consistently appearing in our nuclear samples? For the nuclear insoluble fraction, we identified a total of 2,035 proteins. 28 of those appeared at least once in all 5 conditions, and once the artifacts and contaminants were removed, 23 remained. Of those, 7 were functionally interesting and had antibodies available. For the nuclear soluble fraction, we identified a total of 1,482. 22 appeared at least once in 5 of the 6 conditions, and 17 remained once the artifacts and contaminants were removed. Of those, 4 were functionally interesting and had antibodies available. As 1 protein appeared in both of these lists, this gave us a total shortlist of 10 proteins. We also performed 2 PubMed meta-analyses. So the first used the search terms POMBE and AGO1, and the second used the much broader Argonaut and RITS. From these literature searches, we identified an additional 6 candidates that have been implicated in other RITS systems. These 6 also appeared in our mass spec data. So this gave us a total shortlist of 16. To confirm the shortlist, a series of Western blots and IPs were performed. The first consisted of blotting the lysate with the 16 candidates. The second involved performing an AGO1 IP and then blotting the eluate with the candidates. Here, only 13 of the 16 were confirmed. Finally, the candidates themselves were immunoprecipitated and the eluate blotted with AGO1. Only 5 contained a detectable amount of AGO1. For the 5 that remained, we wanted to see whether there were any previously identified or predicted interactions. To do this, we queried the proteins against a string database, which contains known and predicted protein interactions. From the figure, we can see that AGO1 has previously been determined to interact with only candidate 1, and through candidate 1, candidate 2. Both of these are chaperones that are known to prevent AGO1 degradation. Based on the literature, we actually didn't think that they would be a core RITS component. We thought it was more likely that they would be involved in complex assembly, but we decided to keep them on our shortlist because of their strong connection to AGO1, particularly in our silencing system. Interestingly, there were no known interactions between AGO1 and candidates 3 to 5, which were the 3 that, based on the literature, we thought could plausibly be a RITS component. From here, we analysed the structures and functions of the 5 candidates to see if they contained any similarities to the yeast RITS proteins. CHIP1, if you remember, is the protein that's responsible for anchoring RITS to methylated histone 3 lysine 9. From our candidate list, number 5 was the obvious standout. Like CHIP1, it contains a chromodomain which has been shown to bind to methylated histone 3 lysine 9. Finding a human TAS3 equivalent, on the other hand, was a bit more difficult. TAS3, if you remember, is the bridge between AGO1 and CHIP1. It uses an AGO hook to bind to AGO1's POE domain. This hook motif consists of repeating GW or WG residues. To try and find a suitable candidate, we used AGOs, a server that identifies GW motifs and predicts the likelihood of them interacting with AGO1. As a positive control, we threw in yeast TAS3 and human TNRC6B. Both of these are proteins with well-defined AGO1 binding domains, which are indicated by the top lines with the blue boxes. The bottom lines with the red, yellow, and green are the predicted regions. From the diagram, you can see that the server isn't 100% accurate, but the majority of the medium and high compatibility predictions were correct. When we threw in our shortlist, candidates 1 to 4 returned positive hits. Most had low compatibility predictions, but candidate 3 had a promising medium compatibility, so it became our lead human TAS3 equivalent. To explore candidate AGO1 binding domains, we generated a series of AGO1 GFP mutants. The main panel consisted of single domain deletions. There are reports in the literature that AGO1 must bind to siRNA prior to the RITS complex forming. All of these mutants were carefully designed to include all of the essential binding residues. A smaller additional panel with 2 or more domain deletions was included as a broad screening tool and binding controls. To test AGO1 candidate interactions, the AGO1 GFP mutants were transfected into HeLa cells. 48 hours post-transfection, whole cell lysates were obtained and the mutants immunoprecipitated with a GFP antibody. Mass spectrometry and Western blotting was then performed on the eluate. The heat map here is a summary of the mass spec results. It displays the number of exclusive spectrum counts for all of the shortlisted candidates. In this context, exclusive means that the peptide can only be traced back to a single protein, so there's no ambiguity in terms of protein variants, and the spectrum counts can be used as a rough quantitative measure. From the figure, we can see that there are 3 mutants that stand out: delta PAS, delta mid, and delta N-terminal plus L1. For candidates 1 and 2, the Western blot data supported the heat map. In both data sets, the candidates had reduced binding affinity to the same 3 AGO mutants. Unfortunately, there does seem to have been some confounding of the data. And this is obvious when we compare the mutants with the ones that have been highlighted by the red arrows. So the 3 on the right are missing the same domains as the 2 on the left, but we can see different results. So this tells me that the reduced binding to delta PAS and delta mid is not because of the loss of the domains themselves. So once we remove the confounded data, we're left with 1 mutant, which is delta N-terminal plus L1. And this is a result that reflects the literature. When we look at candidate 3, we can see that there are 4 mutants that stand out. Unfortunately, this data is also confounded. Between these 4 mutants, the entire AGO1 protein is covered. Further, the Western blot for this IP, which I haven't shown, looks completely different. This protein has actually been incredibly difficult to work with in general. In my experience, indirectly immunoprecipitating it just gives you inconsistent results. And in order to get some sort of reliable data, you have to IP the protein itself. From the Western blot data, we can see that the candidate has reduced binding to 4 of the AGO1 mutants, Like the first two candidates, one of them seems to be due to altered epitopes, and this is highlighted by the red arrows. The remaining three, however, support the notion that the candidate binds to the AGO1, L1, and PAS domains. For candidates 4 and 5, we couldn't interpret much from the mass spec due to the low number of spectrum counts. Interestingly, both were detected approximately 4 to 5 folds less than the other candidates, which suggests that they may not be directly binding to AGO1 or are associating with the complex at a different stage. The Western blot data was also difficult to interpret. Both appeared to have reduced binding to the same 4 middle mutants, which suggests that they may be playing a similar role or working in concert. To try to model these interactions, we generated tertiary structures for each of the candidates in FIRE2. FIRE2 is a protein fold recognition server that uses homologous templates to construct the queried sequence. As very little is known about candidate 4, we couldn't generate a tertiary structure. For AGO1 and the remaining candidates, various combinations were submitted to ZDoc to model their interactions. One noteworthy feature is the AGO-L1 domain. L1 is a small bulky protrusion in light grey. It was repeatedly implicated in the candidate interactions. In particular, candidates 1, 2, 3, which we believe are directly binding to AGO1. So this raised the question of competitive binding. When we modelled AGO1 with candidates 1 and 2, we realized that in order for the candidates to bind to both the N-terminal and L1 region, they needed to span AGO1 in a way that partially obscured the PAS region, which is what candidate 3 binds to. When we modelled AGO1 with candidate 3, we realized that no matter how we orientated the protein, it was just not possible for candidates 1 to 3 to simultaneously bind to AGO1. So it seems that we've captured two distinct complexes. As candidates 1 and 2 are known chaperones, our guess is that they form the first complex and serve a preliminary function, perhaps something similar to the yeast preliminary ARC complex, and that once they disengage, candidate 3, our human TAS3 equivalent, replaces them. To try and model a human Ritz complex, we combined AGO1 with our two lead candidates. We didn't perform any interaction studies between the candidates themselves, so the interactions modelled here between candidates 3 and 5 are educated guesses based on the literature. Nonetheless, the proposed complex here can theoretically perform the same functions as the yeast Ritz complex. To test how essential candidates 3 and 5 are for epigenetic silencing, we generated CRISPR knockouts in HeLa cultures. Unexpectedly, the knockout for candidate 3 was quite poor, with only a 14% reduction in protein expression. The knockout for candidate 5 was much higher, with a 90% reduction. These CRISPR cultures were infected with HIV, and 5 hours post-infection, transfected with PRAME. At days 4 and 6, the cultures were harvested and RT-qPCRs for viral gag were performed. From the summary, the first thing we can see is that the mock and scrambled cultures look very similar, and that the PRAME-treated cultures have at least a 100-fold decrease in viral infection compared to the mock and scrambled. These were expected results, and they let us know that our experiment worked. When we look more closely at the mock transfected cultures, we can see that knockout of the candidates alone is sufficient to significantly increase the infection by day 6. This can be explained by the fact that both candidates are natural antiviral factors, which reduce viral infection following HIV integration. And it seems that a few infection cycles are required before we can start to see the effect of their loss. The scrambled transfected cultures looked the same and had the same 2-fold increase at day 6. When we look at the PRAME-treated cultures, we can see a similar trend. Candidate 3 is the obvious standout. The 3.5-fold increase in viral gag at day 6 is significantly higher than the parental and the highest recorded fold change overall. This is pretty promising considering that the candidate 3 knockout culture had only a 14% reduction in protein expression. So taken together, it appears that candidate 3 is required for silencing. Candidate 5, on the other hand, doesn't look like it's required. The 2-fold increase at day 6 mimics the mock and scrambled cultures, but I would actually argue that there is something going on. From HIV infection to integration, there's at least a 15-hour delay. We transfected the cultures 5 hours post-infection. From previous experiments, we know that within 12 hours, Ritz is recruited to the chromatin and starts silencing the virus. So this means that PRAME treatment should be mitigating the loss of the candidate, and that the results should look like the parental. The fact that we can still see that 2-fold difference at day 6 makes me believe that the protein does play a silencing role and that its involvement in HIV pathogenesis is obscuring the results. Now, the work that I presented today is only part of my thesis. I didn't have time to present the entire scope, and one of the major findings that I didn't talk about was the actin cytoskeleton and how we've found that it's required for the intracellular trafficking and nuclear entry of the silencing machinery. So this diagram here combines all the results from my thesis and previous reports in the literature. Starting from step 1, we propose that the duplex siRNA are loaded onto AGO1, and candidates 1 and 2 form a preliminary complex that, similar to the yeast arc, may act as a quality control mechanism, which ensures that loading proceeds for its assembly. Once candidates 1 and 2 disengage, the silencing machinery is assembled and trafficked into the nucleus via the actin cytoskeleton. As candidates 1 and 2 are chaperones, and we found that they also interact with our other candidates, we propose a potential dual role where they may be involved in facilitating complex assembly. Once in the nucleus, candidates 5 uses its chromodomain to anchor RITs anchors RITs to methylated histone 3 lysine 9. As candidate 3 has been linked to chromatin modifying complexes, we propose that it may be what's required to help recruit all the HTACs and other chromatin enzymes for chromatin compaction. Obviously, this model requires further experiments, but it also highlights key steps in the pathway that can be manipulated and enhanced. And importantly, the 5 candidate proteins that I've described today could be used as avenues to help explore these steps. So in conclusion, we've identified numerous promising candidates for human epigenetic silencing, with the top 5 exhibiting functional similarities to the yeast pathway. While we primarily aim to identify the human RITs complex, we seem to have captured additional preliminary elements. Determining the essential AGO1 binding domains was difficult, perhaps due to altered epitopes which are preventing the antibody or candidate protein from binding. Further studies are required to determine the candidate's roles in epigenetic silencing, and these experiments may have to be performed in non-retroviral models to try and avoid potential confounding factors. All of the information gained from these experiments could be used to further enhance siRNA therapeutics, and we can use tactics that have been proven for other strategies, such as delivering the siRNA with the silencing machinery. All of this information can be used to generate safe and powerful therapeutics. And with that, I'd just like to acknowledge everyone who's made this project possible, and in particular my supervisors, Dr. Chantal Allenstiel and Professor Anthony Kelleher. Thank you.
Thank you, Catherine. That was a great overview of your thesis and all the exciting data you've got. All right, don't forget that the question and answer panel is open, so please type in your questions there and we'll address all of them at the end once we've had our second speaker. So our second speaker today is Vanilla Mathivanan, who's an HDR candidate in the HIV biology group led by Associate Professor Stuart Turville in IVPP, and she has also recently submitted her thesis, so congratulations. Vanilla will be presenting her exciting findings that are towards a viable platform to build the generation of a gene therapy to target resting T cells for the use of in vivo gene therapy. Thanks, Vanilla. Thanks, Vanilla. That was great. All right, so please type in your questions for Vanilla. We've got a couple of questions for Catherine, so we'll start the Q&A. The first question was, all right, sorry they missed it, but why is C3 more effective at day 6, but it only had a 14% reduction at earlier time points?
So the 14% reduction actually relates to the level of the CRISPR knockout. So it's just, the thing that I was trying to say was that even though we only knocked out 14% of the protein expression, we still saw significantly less silencing at day 6 compared to the controls. Does that make sense now? Sorry.
That should clarify it. Yes, thank you. All right, so the next question again for Catherine is that the RITs complex is known about and well described in yeast and plants for many years now, but has resisted characterization in mammalian cells. So would you like to speculate why the problem is so difficult to dissect? And if you were starting from now, how would you approach the problem?
That's a really good question. So I think even with yeast, like a lot of the proteins that I described in that yeast pathway they're unique to yeast. They can't actually be found outside of that particular species, or some of them in particular, just S. pombe, just can't be found outside of that one, fission yeast. I think for me, at least, when I was going through my thesis and going through my project, I had a lot of issues trying to identify candidates. I think one of them is highlighted by my CRISPR experiments and the fact that the candidates that I identified also seem to be antiviral factors. So part of it, for me at least, looks like they're also wrapped in other processes, and it's really hard to kind of tease them apart and try and figure out which is due because of process A or process B kind of thing. So if I was starting from now, I would definitely focus on candidates 3 and 5. I think they're probably the most promising in terms of being part of the human-Ritz complex. And I think I would actually like to do some microscopy work on it, which is actually part of the original plan of my thesis, which got derailed from COVID. But I would like to actually see what it looks like if I transfected these cells with my candidates with a fluorescent marker and see whether they co-localize with my siRNA and AGO1 and at what stage, whether it's the cytoplasm or the nucleus. And I think it would also be nice perhaps to do something in just normal HeLa cells without the infection, something without a virus, I guess.
Nice. There's a question here from Alberto. Great talk and amazing work, Catherine. All right, question. Did you look further into knocking out candidate 3? It's interesting that you got such a strong effect despite only a 14% knockout. And he wants to know how you did it. Did you use one guide RNA and bulk transduced cells? And are you attempting to get a clonal knockout? Or is candidate 3 an essential gene? Has it been knocked out previously in the literature? Lots of questions.
So as far as I know, it hasn't been completely knocked out before. And this was actually a Santa Cruz special. So it's not my forte to design guide RNAs. So we thought we'll go with something that's actually been validated. And the Santa Cruz special that we bought, it was actually a mix of 3 plasmids, and each plasmid had its own unique guide RNA. And they didn't tell us if there was any resistance or anything. As far as we knew, there wasn't. There was just a GFP marker for transfection. So what I ended up doing was transfecting my HeLa cells with these plasmids and then getting the lovely Hannah Law to sort them for me based on GFP expression. And even though the majority of them were very high GFP expression, we only had a 14% knockout, which was unexpected. I didn't have time to try and finesse it, so I thought we'll just go with it. It was definitely something to do in future.
Nice. And one quick question from me. So you mentioned earlier on that the Ritz complex has to have a slicing activity. Do any of your candidates have them, and can you speculate on that?
No, none of my candidates have them. There were some candidates that were part of my top 20 list that had the slicing motif, which is usually like a DDH or DDD amino acid triad. And those, they weren't confirmed in my Western blots and IPs, but I think that might just be because I need to tweak them a bit more. It could be that they are incredibly transient, which is why I couldn't detect them by Western blot, but I could by mass spec, which is a lot more sensitive. So I think it's definitely something to follow up in the future.
So with that, I'd like to get everyone to thank our speakers. Well done, guys. Good work, and congratulations to both of you on your submissions. And I'd like to thank everyone for joining us this afternoon and wish you all a lovely day. Thank you. Bye.